And I would like to thank the platform for giving me the opportunity to share I think one of the most important aspect of milk, that is about the exosomes, which is the supposed to be the future of the dairy industry, because the pace is going on in evaluation of various fractionated components of milk in different aspects. So at present, I am working on this milk-derived exosomes, and it's evaluating its properties in different aspects, and not much work has been done in case of indigenous breeds in India, like cattle, buffalo, this Sahiwal cattle, this beetle, goat milk, and Mura buffalo milk. So few works has been done till now, and we are in the way to explore many more. So I welcome all the participants to have a brief idea about milk-derived exosomes and its future. So shall I start, ma'am?
Yes, ma'am, please.
Okay. So we all know about milk. Basically, if we will define it by as a biochemist, it is a liquid or fluid. It is secreted by mammary gland of female mammals, all female mammals, to nourish their young for a period beginning immediately after birth. So, the milk of domesticated animals is an important food source for human, either as a fresh form or in processed form when converted into a number of dairy products like butter, cheese, yogurt, and etc. Mostly, source of milk in Western countries are cow, sheep, and goat, whereas in Asia, including India, there is, in addition to these species, there is also buffalo and camel milk, which is also preferred. Buffalo milk is mostly preferred in India, whereas camel milk is in its way. So, if we'll define biochemically what is milk, it is an emulsion of fat and protein. If we divide its components, there will be fat and there will be protein that is in water, we can say. So, in addition, there is also sugar like carbohydrates and minerals and so many vitamins also. The biochemical constituents in the milk of all mammals are quite similar, but the proportions differ from species to species. So here is it is in tabulated format. The biochemical constituents of milk of different species, including human. So there is cow, goat, sheep, buffalo milk constituents as compared to human. If you can see, sheep milk, though it is not preferred in India, though in many people don't prefer sheep milk, it has not been popularized till now. But if we can see, it has the highest amount of fat content, energy content, protein content, and calcium, phosphorus, and carbohydrates. Though carbohydrate is higher in human, but it has also significantly higher carbohydrate content as compared to other species. So the milk is of each species is a complete food for its own young. Nature has designed it in such a way that it provides all the required nutrient to the young one, to the infant after its birth. In the stomach of the young, milk is converted to soft curd by different enzymes, and that encloses the globules of fat, enabling digestion to proceed smoothly. Lactose or milk sugar is broken down into simple digestive sugars by the enzyme lactase, which is produced in the intestine of infant, and it is gradually absorbed into the body. Then it acts as a source of energy for the infant for every period of time till development of its complete digestive system or other system that can act fully naturally to act itself as a source of energy, as development of all the metabolic enzymes that can produce energy in the body. So we know all about benefits of milk. Still, this is the summarized aspect of health benefits and functional properties of milk. Like milk is little rich in amino acids like tryptophan and cysteine that it prevents growth of harmful bacteria and reduces the risk of infection. It has immunomodulatory effects. It has anti-allergic properties, anti-inflammatory properties, antioxidant. It acts as an antioxidant. In addition, it acts as an antimicrobial and anti-cancerous agents also. It modulates production of various cytokines and other immune system components in the infant or in the adults also. It is used somehow, some cases it is used for treatment of some autoimmune diseases, allergies, and some other immune system disorders. It acts as a better emulsifying and foaming agent in agent. And there are so many bioactive peptides present in milk and whey protein. The main protein component of milk is whey and casein. And when these proteins are digested by different methodologies like enzymatic or microbial digestion or any kind of digestion, so many peptides are developed from this protein contents. So maybe this by protease heat treatment or microbial fermentation, like milk is always subjected to fermentation to production of various milk derived products, and it is usually goes by heat treatment during pasteurization and all, and enzymatic treatment like gastric protease, pepsin, or lactic acid microorganism derived enzymes. Those are present in the intestine in the natural way. So these proteins undergoes various kind of hydrolysis to generate different types of bioactive peptides. When the protein is digested inside the body, some peptides are produced, and those peptides are having active properties like antimicrobial, antioxidative, or anti-cancerous. Then these peptides are known as bioactive peptides. There are so many ways to produce bioactive peptides from the protein content. So, like in case of um, milk proteins with casein and whey protein, they undergo different types of treatments to produce bioactive peptides that has activity like antimicrobial, antioxidative, anti-glycemic. Okay, and some are also having S inhibitor. They act as S inhibitor, acetylcholine esterase inhibitor, along with antimicrobial and antioxidative properties. So these are the peptides. You can see P, E, Q, S, L, A, C, Q, C, L. These are the amino acid sequences of the protein beta-lactoglobulin from the position of 113 to 122 amino acids. Like this from beta-casein, kappa-casein, lactoglobulin. These are the peptides derived which has bioactive properties. Like these specific peptides are reported to be having inhibitory activity on acetylcholine esterase. So they act as somehow they act as antihypertensive agent. Their properties are equivalent to that of the drug captopril that act as an antihypertensive. Mostly it is taken as VP medicine, anti-VP medicine, and milk has these proteins, and from this protein these peptides are derived, and these peptides are having similar equivalent action like that of antihypertensive drug captopril. So these peptides are having anti-inflammatory, anti-obesity, and immunomodulatory activities as reported by so many researchers. So the dose-dependent inhibition on human peripheral blood molecular cells in vitro has been observed due to interference of generation of important T cell activating signals for anti-inflammatory properties. Then alpha S2 casein of milk or milk yogurt would repair the ileum microstructure in rat arthritis group suppressing the inflammatory process. The activities has been observed. Milk in the diet decrease body weight, fat mass, improved glucose tolerance, energy expenditure, and oxidative fibers in skeletal muscles, reduces inflammatory markers, and prevent adipose tissue hypertrophy and hepatic steatosis in mice fed with high fat diet. Along with this, it has anti-COVID properties. Anti-COVID properties like it controls the expression of interleukin C in interleukin 6 in COVID-19 patients. It has been observed, and these are the peptides that is responsible for or that act as interleukin 6 inhibitor in the COVID-19 patients. Apart from the peptides, there is oligosaccharides, there is milk sugars. So lactose is the major milk sugar in the milk. Lactose is composed of glucose and galactose. These are the monosaccharides, and lactose is basically a disaccharide. It is uh, it is supposed to favor absorption of different minerals like calcium, magnesium, and phosphorus. Along with it, helps in utilization of vitamin D. These oligosaccharides also enhances sensory properties. They have prebiotic properties, probiotic properties, and drug delivery capacities. So these are some of the natural oligosaccharides present in milk, apart from the what we say lactose, like 3-galactosyl lactose, lacto-N-hexose, N-acetyl glucosamine lactose. So these are the derivative of lactose. Like that, some acidic oligosaccharides are also there. Sialyl lactose, glucolyl neuraminyl lactose. These are different types of lactose derivatives that is present in milk that is supposed to have different prebiotic and probiotic properties. Apart from this, carbohydrates and proteins, there are some cells are also present in milk. We can say different monocytes, macrophages, neutrophils, cytotoxic T regulatory cells, NK cells, beta cells, etc. They are responsible for balance between infection and making the animal or human tolerance to harmless environmental compounds. So they reinforce and educate neonatal immune trajectories, allowing the best protection to the newborn. They produce bioactive molecules like lactoferrin, lysozyme, oligosaccharides, and cytokines and others that help the neonatal immune system to strengthen up. And the beta cells, they produce immunoglobulins that is highly responsible for development of immunity in the newborns. Milk-derived beta cells are active cells with high level of CD38 expression and low expression of complement receptors. That is a feature of plasma cell. Soluble milk immunoglobulin A also have antimicrobial properties and are present highly in milk. It is transported through milk from mother to the infant. This immunoglobulin A could inhibit pathogen binding by complexing antigens and taking them up by the intestinal dendrocyte cells, which will then process and present the antigen. Apart from this, there is immunoglobulin M, antimicrobial antibodies. These are also found in milk that mediate cytotoxicity. The next one is macrophage. This is involved in immune protection of the infant by high phagocytic role, and these are secreting granulocyte macrophage colony stimulating factor, that is EMCSF. In the presence of exogenous interleukin 4 alone, it differentiates into CD1 plus dendrocytic cells, so their ability to stimulate T cells. So it is responsible for the anti-inflammatory properties, so protecting the infant against mucosal inflammation. It plays a crucial role in gastrointestinal mucosa and are involved in the maturation of immune system of the nursing infant without causing any inflammation. It helps in development of innate immunity, but also it triggers high antigen specific T cell development in the intestinal mucosa of the newborns. The next one is T lymphocytes. As we know, T regulatory cells are the main player for providing tolerance to the newborn. So these CD127, CD25 positive cells are the main T regulatory cells found in the colostrum, then peripheral blood, who is showing passive transfer of tolerance mediated by memory cells and is positively associated with vaginal delivery. The T regulatory cells play beneficial role in the newborn by lowering the inflammation in the gut mucosa and reducing the risk of developing allergy, asthma, obesity, and diabetes type 1 during the certain long-term life of the infant. So these are all the cells present in milk. We usually conventionally we know that immunoglobulins are transferred from the mother to the child to the infant through the milk. But apart from this immunoglobulin, there are also different immune cells that is secreted in the milk and that is also transferred from the mother to the infant through the milk, and it helps in development of intestinal immunity and gradually the complete immunity or defense system of the infant. Gradually it helps in development of the immunity. So now coming to the vesicles, extracellular vesicles, we have discussed about the cells present in milk. So there are, apart from these cells, these proteins, peptides, this carbohydrate content, mineral and vitamins, there are some other micro level bodies, we can say. These are present in milk. These are known as extracellular vesicles because these are not a cell, but these are derived from the cells. That's why it is extracellular vesicles. And these are vesicles means these are round-like structure containing some biological components. So we'll be discussing about that. So these extracellular vesicles, these are produced by all living cells and they have specific role in cell-to-cell communication. So these are ubiquitous in living cells and it is having physiological and pathological significance also. So we can see there are different types of vesicles, like this is the first one is exosomes, second one is some apoptotic bodies, microvesicles and exomeres. These are all derived from cells. Cells and according to the size, there is the variation. Like exosomes, that is 70 to 150 nanometer in size, whereas microvesicles, that is 100 to 1000 nanometer. Apoptotic body is a little larger, that is 500 to 2000 nanometer, whereas exomeres are the smallest one, smaller than the exosomes, that is less than 50 nanometer in size. And there is also variation of contents. Like exosomes contain proteins, lipids, and nucleic acid. It has specific membrane, and its main function is intercellular communication. It originated by endosomal pathway inside the cell. Whereas microvesicles, they also contain protein, lipids, and nucleic acid. They are also have specific membrane and it contain RNAs, proteins, lipids inside it. It is also responsible for intercellular communication. It is developed by budding. Whereas exomeres, which are the smallest one, these are not defined by any membrane, but there is no lipid bilayer, but it contains few proteins, peptides, lipids, and nucleic acid. It's also responsible for intercellular communication. Whereas apoptotic bodies, these are the largest one. They contain some nuclear fragments and organelles also, as it is related to apoptosis process, means cell death process. This is developed by gradually shrinkage or membrane blebbing during apoptosis. So our focus is on exosomes. These are membrane bound vesicles. These are the size is 40 to 100 nanometer, mostly average 100 nanometer or less than 100 nanometer. They have specific role in communication, cell to cell communication, but they carry some bioactive substances like proteins, RNA, DNA. Proteins also contain peptides out of RNA. miRNA is the most important one that is present in exosomes that has therapeutic properties. DNA is also present. This uh, in the electron microscope, we can see the size is average 100 nanometer. These are membrane bound globular structure, and it is a fluid. It surrounds some of the fluid inside it, which contain these bioactive substances like proteins, nucleic acids, miRNA, DNA, extra lipids also. And this, as it is surrounded by some membrane, this is there are some membrane markers on it. So these are the membrane markers like CD81, CD9, 82, 63, like that. So this is for recognition or confirmation of these exosomes. So these exosomes are cell-derived bodies. So it is present in all types of fluids in an animal body or human body. Like it is present in blood, urine, saliva, milk, amniotic fluid, semen, and ascites, cerebrospinal fluid, etc. This is present in all the body fluids. It is responsible and it carry the bioactive substances specific to that fluid, specific to that system, indicating any damage or any functional activities related to that system. So these exosomes are, this is about the milestones in the discovery of these exosomes. These are identified 1983 from the SIP reticulocytes. It is first defined as intraluminal vesicles released from the cell by innovative studies from the Johnston and Stull laboratories. And gradually, the journey goes on. First, it is derived as a cell-derived particle, then matrix vesicles, then microvesicles. Then in 1981, there is arrival of these exosomes. Then there is studies goes on increasing related to the exosomal functions, like its secretion, its mechanism of production, this function, then derivation from different types of cells like immune cells, cancer cells, then what it carries, what are the cargoes it contain, like that. So from 1974 to 2007, like this, the journey goes on. And in 2007, it is the exosomes derived from milk was started, and its functional studies started from milk. So exosomes are present in beast milk, and it was first identified by electron microscopy exhibiting typical exosome characteristics and protein markers in 2007. These milk exosomes are supposed to be found to be rich in membrane glycoproteins, fat global epidermal growth factors, and that is prominent in the lactating mammary gland, as discovered by in 2006 by Nakatani. So here we can see this is the mammary gland. This is the system, ducts, gland system, through which the milk is released to the teat cistern and teat canal. It is released out, and these are the cistern. Okay, so this milk is secreted from the mammary gland from the this epithelial cells. These are the alveolar epithelial cells. The milk is synthesized here and secreted into the lumen here. So milk is but basically a cell derived product. And along with this milk, there is also this mammary gland is also surrounded by lymphatic vessels, and from the lymph, the different types of immune cells that also secreted into the milk, and combinedly they form the milk what it comes in its pure form. So this is the alveolar epithelial cells with the brustbundle epithelium, and this is the process of exosome synthesis from the alveolar epithelial cells. So this exosomes synthesized from this cell comes into the milk in all the species, maybe it camel or pig or sheep or buffalo, bovine or human milk. It secreted into the raw milk. It is present in the processed milk. It is also present in the colostrum. Then, so this is the vesicle with its this exosomal vesicle with its all its markers and its cargo, its content, like different types of metabolites, proteins, nucleic acid, lipids are present in it. So these are some of the listed milk derived miRNAs, microRNAs. Uh, we ingest it, uh, these exosomes along with the milk, and it goes to the intestine. It is absorbed in the intestine. Then it is distributed to the body, entire body system, and its therapeutic effect is acted throughout the body. So it is secreted primarily by the bovine or mammalian mammary alveolar cells. So this is the cell. This is the picture depicting entire process of synthesis of these exosomes. This is the cell membrane. This is the extracellular space like that. In the process of endocytosis, some membrane component is internalized to form vesicle-like component inside the cell. This uh, goes to the step of early endosome preparation. Then through the Golgi bodies or through this ESCRT system, there will be incorporation of different markers, tetraspannins, or incorporation of different components, proteins, lipids, some miRNAs, nucleic acids inside the endosome. Then there will be formation of intraluminar vesicles, that is again transported to the plasma membrane. Then there will be docking and fusion with plasma membrane. Then by the process of exocytosis, these exosomes are released to the extracellular space. This is the whole mechanism of formation of these exosomes in any cells. So in the bovine or human or any species, any mammary alveolar cells, the process goes on like this. So as it is derived from the membrane, there is a lipid bilayer in its confining its structure with different markers like tetraspannins. There is also antigen presenting molecules like MSC class 1 and class 2. Then signaling receptors like TNF receptor, TFR receptors like that. There is in the membrane, there is present lipids, cholesterol, ceramides, sphingomyelin, etc., which is the normal membrane components. There is glycoproteins. They also act as some kind of receptors here, or this is required for this intercellular communication, etc. There is adhesion molecules, etc., in the membrane. And inside the membrane, there is different types of heat shock proteins. There is cytoskeletal proteins, membrane transport proteins, then growth factors, cytokines, nucleic acid, mRNA, miRNA, etc. In its content. So these are isolated by different processes because these are very minute particles. These are in the nanometer range, so there is this cannot be isolated by. So mostly it is isolated by ultracentrifugation, that to differential ultracentrifugation. Differential means we have to remove all the components one by one, like proteins, carbohydrates, then large vesicles, then small vesicles, like that at different speeds. We have to remove the content step by step, and finally at the range of around 1 lakh RCF, these exosomes are pelleted, and these are extracted from the milk or any kind of body fluid. So this differential ultracentrifugation process is the gold standard for its purification, but it takes much more time, and somehow it impacts its integrity, its membrane contents also. So gradually there is development of size exclusion chromatography that has been developed nowadays for isolation of these exosomes from any kind of fluid. And this is the process step that is required for followed for isolation of exosomes from milk, like raw milk. It is ultra centrifugated at first at 12,000 G for 4 degrees Celsius to remove the fat and cell debris. Then there will be formation of skimmed milk. Then again there will be centrifugation at 1,50,000 G for 2 hours. The exosome pellet will be collected at the base. Then that is transferred through the column for size exclusion chromatography. Then in different fractions it can be collected. Then these exosomes are characterized by the processes like because we can see it in bare eyes, so we have to follow for this transmission electron microscopy for characterization, then nanoparticle tracking analysis, then dynamic light scattering, flow cytometry, mass spectrometry. These are the high-end techniques for characterization of these exosomes. So as it contains different proteins, different markers. So these have specific properties, or these are specific functions or applications like intercellular signaling, immune response, modulation, inflammatory regulation, maintenance of cellular homeostasis, therapeutic, cargo delivery, or diagnostic markers like that it can act. So it has a role in maintaining or development of intestinal structure or defense system, immunity in the infants. It has a role in metabolism. Also, there are so many miRNAs that regulate different processes of metabolism in the whole body, starting from the infants to grown-up situational condition. Then it also helps in development of nervous system, neuronal parts also. In the intestine, most of the studies has been done its impact on intestine first. So in the intestine, it these milk exosomes they survive the digestive processes, means they are survived from the this enzymatic digestion that occurs inside the intestine. They are absorbed into the through the intestinal epithelial cell to the blood circulation and distributed throughout the body. They are assimilated via endocytosis in the intestinal cells. They help to combat any kind of oxidative stress or intestinal injury or maintenance of the system. Then they are tolerogenic to the immune response also. So they also help to protect against inflammatory bowel disease, NEC, or any kind of malnutrition, because it has some of the metabolites, peptides, and miRNAs, etc., that help to combat these processes. So gradually after ingestion and assimilation throughout the body, it is responsible for various functioning like osteoblast proliferation in bone, then expression of osteoblast related genes, then it increases the bone mineral density in trabecular and corticular tibia like that. As we know, milk contain calcium and it helps in absorption of vitamin D. But there are so many microRNAs that helps in this development of the bone mineralization, etc. So maybe this microRNA is present in the these exosomes that is helpful for the gene expression and mineralization process and increasing the density of bones, osteoblast proliferation like that. In case of muscles, it increases muscle protein synthesis, and that has been studied in vitro and in vivo. The modest effect on gene expression and amino acid content has been observed, whereas no significant effect on muscle strength has been reported. So through the process of angiogenesis, then osteogenic differentiation, mineralization, like these are the RNAs, miRNAs, and these are the transcription factors and the pathways responsible for these processes. So these exosomes help in stimulating these factors that helps in this angiogenesis or osteogenic differentiation, mineralization, like that. These exosomes are supposed to cooperate or help or increases the expression profiling of these factors that help in functioning in this aspect. Coming to the immunity, there are so many immune-related miRNAs present in the milk exosomes. Few are enlisted here, like this miRNA 148, 152 family, or 148A, or LET7 miRNA. That these are responsible for inhibiting TLR-mediated expression of MSC2 and cytokine production. It's regulate the function of P and T lymphocytes, TLR4 signaling, etc. And the protein content is responsible for Th17 differences. So all these factors are responsible for overall development of immune system in the infant to the kid. Further, it helps in development of nervous system as it can cross the blood-brain barrier. So it has been studied like mice fed on exosome deficient diet. There is impaired cognitive performance, whereas if it is fed with exosomes, then it has enhanced spatial learning and improved memory. That has been seen in case of laboratory animals like mice. So these exosomes also helps in maintenance of gut microbiota. It increases growth of E. coli and L. plantarum commensal stage in the gut, in the intestine. There is differential abundance in 3 phyla, 7 families, and 52 operational taxonomic units in exosome sufficient and exosome depletion that are studied by Joe in 2019. So overall, it has different functions like drug delivery, imaging, and this has markers of various disease. This has been used as wound healing process and in therapeutic in different disease conditions. So recently, these exosomes are used as a vehicle to deliver drug, as it is the particle size in nano range, so it mimic the action of nanoparticles. So because of its nano structure and lipid layer it contains, so it has a low immunogenicity as compared to the nanoparticle, the synthetic nanoparticles. It has minimum side effects, and it has low toxicity, high stability, and it can overcome the brain barrier, blood-brain barrier. So it can act as as a nano vehicle for delivery of drugs. So many studies are going on in this aspect. So it has additional advantage compared with lipid nanoparticles and culture-derived exosomes. These milk-derived exosomes have specific properties for drug delivery because it can be it is orally ingested and it can sustain the digestive enzymes. It is stable in the acidic medium. It can protect the cargo, that is the drugs or any other content that we are supposed to deliver. It has high oral bioavailability. It has low immunotoxicity. So it is most preferred nowadays as the delivery vehicle for different kinds of drugs, including anti-cancerous or diabetes drugs like that. So these are the promising drug carrier for treating diverse diseases, highlighting their therapeutic utility. So these exosomes are the delivery vectors for small RNA, siRNA, small interference RNA delivery to silence specific genes also, like VGF, EGFR, AKT, etc. So these are some of the drug needs some of the contains that is used, that is delivered along with the exosomes like curcumins, anthocyanidins, and paclitaxel is the drug. These enhances its therapeutic efficacy and stability, and threefold increase in transport across the CACR2 cell monolayer as studied in vitro in cell lines. So these exosomes are having good properties to deliver these agents, these bioactive compounds at the supposed places, and it can increase the bioavailability in the body also. So anthocyanidins, these are also used or studied to reduce the tumor growth in ovarian cancer mouse model. Then paclitaxel is a drug that has been delivered, that is having anti-cancerous activity. So that has been delivered through exosomes, increasing the its therapeutic efficacy as compared to the free drug delivery via intraperitoneal routes. So in this process of drug delivery, this is the whole schematic presentation of how these drugs can be delivered. Like these are the exosomes, and we supposed to deliver some drug or other cargo materials like siRNA or miRNA or any other kind of bioactive components, immunostimulating agents. So you can add to the exosomes the way it acts like the nanoparticles. There will be incubation or sonication or electroporation. Like these are the processes by which these cargoes are incorporated into these exosomes. Then these are orally delivered. So this is a scheme, complete scheme, that is the milk isolation of exosomes, then targeting, loading of therapeutic agents, then delivery of this loaded exosomes into the cell culture or the organism. It can be delivered to the cell and to the organism, then to see the visualize the effect. So this is the whole process. So we can go for imaging by various method to study the its delivery properties, how it deliver the cargoes at right places, or how it acts is supposed to affect, how it is acting, its complete functioning. So apart from that, there are the miRNAs which are present in the exosomes, that is supposed to be marker of various diseases. In case of disease conditions, there is alteration in the cells, and that alterations are also reflected in the exosomes in terms of because of which these exosomes also act as biomarkers. So they are the biomarkers for quality control of raw milk or milk related products. So as nowadays most of the researchers are focusing towards miRNA and its marker properties, marker as diagnostic, marker as therapeutic effects, etc. So these are the miRNAs enlisted that act as biomarkers for quality control of raw milk or milk related products. So they are identified specific miRNAs related to bacterial infection like BTA, MIR 142A, MIR 223 in milk from cows challenged with Staphylococcus aureus. And also in case of cancer, ME21, that is known as OncoMEAT because it is the marker of cancer. So this is also observed or found in this exosomes of milk in case of cancer also. So these two microRNAs are reported to be the marker candidate for mastitis in case of bovines. So we can detect bovine by this miRNA specific detection techniques, or we can also, it can also help in therapeutic purpose also. We can develop some miRNAs to some this nucleic acid components to block this miRNA development that can act as that is the candidate for this mastitis. So it can also be used for therapeutic purpose. So camel milk derived exosomes has been these are the these exosomes derived from camel, cow, or yak milk has been studied in its anti-cancerous properties. Like they are helpful to reduce cancer metastasis. They increase the apoptosis markers, leading the cancerous cell to death. They attenuated oxidative stress. They have modulate the inflammation related gene expression. They are the carrier for delivering chemotherapeutic agents. They have superior efficacy in inhibiting lung tumor xenograft as it contain MSC2 in its content. They are also responsible for alleviating hypoxic conditions and that is the characteristic of tumor microenvironment. So it helps in cancer treatment. Nowadays research is also going on for this its own healing properties. Like milk derived exosomes, it contain TGF beta, or it modulate the SMAD signaling pathway, upregulating TGF beta 3 expression. That is helpful for wound healing. So during this process, it reduces the inflammatory markers, reduces oxidative stress, increases the antioxidant capacity, improve the skin elasticity, like that. So these are the summary of some of the therapeutic application of exosomes. How this is used, this exosomes can be used as adjuvant in diagnosis of tumor immunotherapy. This they act as in wound healing and reducing inflammation, and is cardioprotective agents like that. It is versatile in nature to help any kind of conditions. So overall, uh, milk contain these exosomes having high therapeutic and diagnostic values. But there are some challenges like isolation and storage, because commonly we cannot isolate these exosomes from milk. We need high-end instruments for this isolation and characterization, for its preservation also. And researches are going on how to include it in the medicinal in the drug delivery agents in a better way. So there is challenges in isolation and storage. There is cryopreservation using PBS, but freezing thawing cycle may damage its exosomes. Then optimal storage pH and cycle number are critical for maintaining exosome integrity. So we cannot use this exosomes normally, but we are trying to find out some processes to extract high quantity of these exosomes from milk, different processes, and to increase its storage properties. And its future lies in enhancing the delivery system of drugs or any other cargo or any other miRNAs like that. And different diversified bioactive compounds can be included in milk exosomes for its ultimate functioning. It has a role in perinatal health and immunity, and mostly it has potential therapeutic application in infant nutrition. So these are all the future aspect of these exosomes and its activities. So we hope this is going to be the future of this dairy industry, because nowadays research is going on this exosome isolation, purification, preservation for future aspect, for its utilization in various diversified fields. Mostly it has antimicrobial, antioxidant, anti-cancerous properties because of the miRNAs and peptides present in its content. And in future, this is going to be the best delivery agent as compared to the nanoparticles, synthetic nanoparticles, so that different drugs can be and loaded to these exosomes and can be given as in the form of medicine. So these exosomes can also reduce the dose of drugs that can be used for treating any disease. So I hope this PPT will be useful for all of you, and this information will help you to explore yourself in this way. Thank you all.
So thank you, ma'am. Now participants are requested to ask any query related to this PPT or this presentation.